Modern humans spend up to 90% of their time indoors. We live and work in sealed, climate-controlled environments that frequently suffer from poor ventilation. Trapped inside those spaces is nitrogen dioxide, or NO2. It is a highly reactive, invisible gas linked to respiratory illness, and it steadily accumulates in the rooms where we sleep and work. To counter this indoor threat, we have to examine the molecular pathways plants use to extract gas, and then apply those survival mechanics directly to the way we care for our indoor houseplants. Houseplants are highly adapted chemical processing factories. Microscopically, they work continuously to pull specific gases from the air to sustain their own growth. Solving our modern indoor air crisis requires understanding how these biological systems survive in some of the most suffocating natural environments on Earth. Consider Hygrophila difformis, a plant that lives its entire life fully submerged underwater. This simulation shows the plant's core physics problem. Gas diffusion in water is 10,000 times slower than in air. Second is a chemical hurdle. Underwater, bicarbonate is the dominant carbon source, but membranes are 10,000 times less permeable to it than CO2. Passively waiting for gas to diffuse across that barrier is a death sentence. To survive, the plant engineers a highly active mechanical solution to pull carbon inside. H. difformis survives using a precise three-step sequential pipeline to actively overcome water's physical limitations. Step 1, outside the cell, carbonic anhydrase hydrates CO2 into bicarbonate. Step 2, specialized transporters grab that bicarbonate, pumping it across the impermeable membrane. Step 3, an internal carbonic anhydrase directs the carbon straight to the chloroplast for photosynthesis. Biologists mapped this pipeline by applying chemical inhibitors to the plant. When they blocked the external enzyme or disabled the membrane transporter, the entire system halted, proving these steps must happen in a specific order. Ultimately, plant cell membranes function as dynamic, multi-stage industrial pipelines, perfectly tailored to extract exact molecules from hostile surroundings. When submerged, H. difformis dynamically rewires its own genetics, upregulating thousands of distinct genes to build out this carbon extraction infrastructure. When the water pH drops, the plant deploys specialized aquaporins, tunnels highly permeable to CO2, maximizing intake under new conditions. Plants constantly monitor their immediate environment and adapt their molecular pumps to match the exact chemical composition of the water or air around them. Knowing how aggressively a plant will fight for gas underwater, we can test what happens when we place these molecular engines into polluted human environments. Researchers exposed the peace lily, ZZ plant, and Dracaena to 100 parts per billion of NO2. All three reduced the pollutant, but Dracaena fragrance was most effective. In a standard 15 cubic meter office, one potted plant removes 0.62 parts per billion of NO2 per hour. Potted plants are utilizing ancient evolutionary survival mechanisms to physically scrub the modern air we breathe. However, a biological engine requires specific environmental fuel to operate its chemical pumps efficiently. Laboratory results show two critical variables maximize NO2 removal. The growing media must remain wet, and leaves need adequate lighting, measured at around 500 lux. You can maximize this natural filtration system by selecting a proven species like Dracaena, providing 500 lux of light, and maintaining moist soil. Operating houseplants as complex biochemical engines allows anyone to affordably and scientifically clear the air inside their own home.